Welcome to part 13 of this video on the immune system. Okay, so we've now discussed VDJ recombination of the alpha chain locus. What we're going to move on to now is VDJ recombination of the beta chain locus. So this is going to be the mechanism by which all of our lymphoid progenitor cells are now going to get their own unique beta chain, which they'll then produce lots of copies of and put with their alpha chains to make a complete T-cell receptor. Okay, so let's discuss beta chain VDJ recombination. Okay, so firstly, what chromosome then is the beta chain locus present on? So the beta chain locus is present on chromosome 7. So again, you'll have two copies of the beta chain locus because you have two copies of chromosome 7. And just like with the alpha chain locus, where we used one of them in VDJ recombination, and then the second was there if the first one went wrong. So if the first VDJ recombination went wrong, then you went to the second copy of the alpha chain locus on the other chromosome 14, and then you performed VDJ recombination there and hoped that that one would go well. It's exactly the same principle for the beta chain loci. You will perform it on one. If that goes well, then of course you just won't bother performing it on the other, and the other beta chain locus isn't capable of actually doing anything. It won't be transcribed and translated into beta chains, so it won't make any difference that it's there. So you just leave it be. Whereas if something goes wrong with the first VDJ recombination, you can then go to the second beta chain, although as we'll see it's slightly more complicated in the case of the beta chain locus. Each beta chain locus on each of the chromosomes actually has two separate tries. It has round 1 and round 2, and only once round 1 and round 2 have failed on your first beta chain locus will you then go to the second beta chain locus. But don't worry, I don't expect you to know what that means yet. I'm about to explain it. Okay, so to start with then, what we're going to do is we're going to have a look at the germline form of the beta chain locus, which will of course be the form of the beta chain locus or in the lymphoid progenitor cells when they've just arrived in the thymus. So I'm going to start by drawing out the beta chain locus in its germline form. So we'll do it up here, and I hope that will be enough space. So this of course is representing once again the double-stranded DNA. Okay, so we're going to see that this is very similar to the alpha chain locus. We've got loads of gene segments. The difference here is that we're going to now have some diversity gene segments as well as the variable and joining gene segments. The other major difference here is that, as I've already said, we're going to have two separate attempts, and I will explain what that means. There's going to be a way for each of the beta chain loci to give you two separate attempts at actually performing VDJ recombination and getting a successful final gene that you can actually use to make your beta chain. Okay, so the other thing that I need to say before we start actually drawing this picture of the beta chain locus in its germline form out is that I'm not going to give you numbers in the way that I gave you numbers for the alpha chain locus. In the case of the alpha chain locus, I told you there were 45 variable gene segments and 55 joining gene segments. I'm not going to give you specific numbers here now. One, because I don't know them. Secondly, because the concepts are far more important than the specific numbers. And thirdly, because the numbers are liable to change. So I just want you to get the concept here rather than the specific numbers. The specific numbers were there previously to give you a concrete example to make sure that the message is nailed home, but I'm hoping now that you have some understanding of the idea and that we don't need that sort of level of concreteness now. Okay, so onto the beta chain locus then. So the beta chain locus in its germline form is what I'm drawing out at the moment. Firstly, you'll have a section of variable gene segments, just like we had in the alpha chain locus, so loads of variable gene segments. And as I say, I'm not going to give you the specific number that you have here because I don't know the specific number. Okay, so you have a bunch of variable gene segments, however, and the number of them will be between 1 and 100 or some number around there. Okay, so these are the first set. This is a first set of variable gene segments here then. So a whole bunch of variable gene segments, and of course to actually produce the beta chain variable portion, you only need one of these variable gene segments. You don't need all of them. Then what you're going to have next up is a diversity gene segment. You're only going to have one there. Okay, so this is just going to be called the D1 diversity segment. And I'll just color that in yellow. And to produce a beta chain variable portion, you do actually need to include a diversity gene segment. So if we've only got one there, it looks as though we're going to have to include that one. Okay, but we'll come back to that in a moment. At the moment, we're just studying the germline form of the beta chain locus. We'll discuss VDJ recombination in a moment. Okay, next up. Next up, we're going to have loads of the joining gene segments. So I'll put these here. So a whole cluster of joining gene segments is here. And I'll colour these joining gene segments in orange. So these are all joining gene segments, 1, 2, and then it'll go on to some sort of number here. Okay, so these are all joining gene segments. Right, so we have a selection of joining gene segments to select from as for our choice of joining gene segment. Then, of course, you'll have a constant region. Here comes the constant gene segment, which will code for the constant region of our beta chain. However, this is where it's going to become a little bit more complicated, because this is actually the first of 2 constant gene segments. So this one is actually only going to be C1. Okay, now where's C2 going to be then? And what's the function of C2? Well, I'll come to what the function of C2 is later. Let's just see where it is firstly. So it's not just after C1. Instead, you have a whole bunch of other things after C1. Before you get to the C2. So you're actually going to have another diversity gene segment. So another option for the diversity gene segment is here. Here is D2 in yellow, like so. So another diversity gene segment there. So this one performs the same role as this one, but will be slightly different. And then you're going to have another huge great array of joining gene segments. So loads of possible joining gene segments that you could use for your joining gene segment in making the variable portion of the alpha chain. Okay, so these are all, the beta chain we're now talking about. So these are all joining gene segments again. So another great big cluster of joining gene segments is here for you to choose from. And then finally you'll have the other constant gene segment here. So this region of DNA that I've now put a box around there, that's the second choice of constant gene segment. Again, I'll colour that one in green here. So this is constant 2. Right, so this is what the germline form of the beta chain locus actually looks like on your chromosome 7s. This is what it will look like in these lymphoid progenitor cells that are just arriving in the thymus gland. So what's now going to happen to actually produce us the beta chain locus that we can actually use to produce our beta chain? Well, of course, VDJ recombination is going to occur. And the way it's going to work is you're going to start off with this up here. So you're going to start off with, ignore all of this back here initially. Just look at this starting bit. The first selection of variable gene segments are one diversity gene segment there, this section of J segments that we've got to choose from there, and then this constant gene segment that we've got there. Ignore all of this initially, and because that's what the cell will do initially. It ignores the existence of this, and it just imagines that this exists. And of course, that bit is perfectly simple. What you're going to do is you're going to pick a variable gene segment, you're going to pick a joining gene segment from these huge selections that you've got here. What you're then going to do is you're going to chop out all of the DNA between your chosen variable gene segment. And again, because it just is easy to draw, let's imagine that I have picked this variable gene segment here, the second one again. What you're going to do is cut out the DNA from this chosen variable gene segment up to the diversity gene segment here, because you've got to include the diversity gene segment. You're going to cut out all of that, and you're going to join your selected variable gene segment to the diversity gene segment. You're then going to do the same thing for the diversity and joining gene segments here. So you'll pick your joining gene segments. Let's say we've picked this second joining gene segment again because it's just so simple, and you'll cut in front of the joining gene segment that you've chosen, you'll cut at the end of the diversity gene segment that you've chosen, and you'll cut all of that DNA there out, and you'll bring this and join it to the diversity gene segment. So what you will then end up with is this is the final result. And I'll leave this bit off for the moment. I'll just show this bit just to make the picture simpler. So here is the first variable gene segment. Here is our chosen variable gene segment, which was the second one. Here's now the diversity gene segment, now stitched next to our chosen variable gene segment. And here's our chosen joining gene segment, stitched next to our diversity gene segment. Then we'll have all of the other joining gene segments after it, and then the constant gene segment here. So let me just colour this in. So again, here's the first variable gene segment. Here's our chosen variable gene segment. And let me just stress that in my pictures, I've always chosen the second variable gene segment. If you had chosen, let's say, the 30th variable gene segment, you'd have 29 more variable gene segments in front of you still. Okay, in this case, you just have one because I've always just chosen the second variable gene segment. Okay, then after that, you'll have the diversity gene segment, which I'll colour in there in yellow, and then your chosen joining gene segment here in orange, then all of the other remaining joining gene segments after it. Okay, so all of them. And then here is our first constant gene segment, like so. Okay, and again, the way that you will get diversity is through combinatorial diversity. That's obvious, that you have all of these different combinations of variable and joining gene segments that you could produce. And secondly, you'll also get diversity by junctional diversity. Okay, so these places where you perform these cuts were not set in stone. There were different places that you could have performed the cuts, and that means that in between the variable gene segment and the diversity gene segment, and in between the diversity and joining gene segment, there is a variable amount of DNA. There are different possibilities for how much DNA you can have in there, and that will also help to produce us diversity in this portion that's now going to code for the variable portion of our beta chain. Okay, so if this goes perfectly successfully, what will now happen is, of course, you will transcribe all of from here all the way down to here, and this is the piece of mRNA you'll get. So you'll get, here is the variable gene segments bit of RNA, here is the diversity gene segments corresponding piece of RNA, here is the joining gene segments piece of RNA, and then of course you'll have all the other joining gene segments there in between, so dot 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 there. And then finally, here is the constant gene segments piece of mRNA. So of course, again, this is the pre-mRNA, and what will now happen is a splicing process which will splice out all of this RNA here. Oh, and by the way, there should be an intron in between that final joining gene segment and the constant gene segment. So imagine there's a clear bit there. You'll cut out all of that up to the beginning of the constant gene segment, and you'll then get your mature mRNA, which will look like this. Here is the chosen variable gene segment. Here is the chosen, well, not the chosen, but the only choice of diversity gene segment. Here's the chosen joining gene segment, and then finally there is the constant gene segment. So this is then now the mature mRNA, and that can now be used to produce your beta chain. So this will undergo translation now, and we'll actually end up with our final beta chain. And again, I'll stress that these portions up here, the variable gene segment, the diversity gene segment, and the joining gene segment, they'll code for the variable domain of the beta chain, whilst the constant gene segment will code for the constant portion of the beta chain. Okay, right. So that's what happens if everything goes to plan. So what is all of this bit here for? This is for round 2. So if things don't go to plan, and what do I mean by things not going to plan? Well, for instance, if you end up because of the junctional diversity, remember junctional diversity comes at a price. It might mean that the diversity and the joining gene segments are going to be read out of frame, and that's a disaster. That's not functional. It won't produce you a functional T-cell receptor. That's too much variation. Okay? If that happens, we need to change something. So what do we do? So if the first attempt was unsuccessful, what you can then do is use this portion of the beta chain locus here. So let me now just actually put this on to this picture. So after your first VDJ recombination, initially in the picture I just drew up to here, but let's now actually go a little bit further, and I'm just at the moment wasting time writing that, well, labeling this arrow as VDJ recombination. Okay, so let me now add on the bits that remain behind here, and we can now see what their function is going to be. So here is the second diversity gene segment, which I'll just colour in here in yellow. Okay, then we have another whole array of joining gene segments to choose from. So loads of possible joining gene segments here. So a great big array of joining gene segments, and then we have the second constant gene segment here, C2. So let me firstly just colour this in. So this is an array of joining gene segments, and I'll just colour all of those in orange. So the first one, second one, and then again I haven't bothered to give you the number, but there will be a certain number of them there, and these are all joining gene segments. And then we have our second constant gene segment right at the end here. So what can now happen if the first VDJ recombination was unsuccessful? So if this is rubbish, if the diversity or the joining gene segments are being read out of phase, we need to do something. So what we do is we now cut out all of the DNA from our chosen variable gene segment all the way up to the next diversity gene segment. So we're going to still use our chosen variable gene segment. We stick with our chosen variable gene segment, but we now say, okay, this bit went rubbish. So now what we're going to do is we're going to go to round 2. We'll cut all of this out and we'll stitch it to this second diversity gene segment. Then what we'll do is we'll choose a new joining gene segment from one of these, and again, because I'm boring and want an easy picture to draw, let's just say I'm picking the second joining gene segment here. We would then cut out all of the DNA between this diversity gene segment and the chosen joining gene segment. And what would we then end up with? Well, of course, what we'd finally end up with is something that looks like this. So this second VDJ recombination. So we'd still have that variable gene segment that's right at the front. Here's our chosen variable gene segment, so we haven't lost the first one. So here is the first one here. Here's the second one, the chosen one. And then we'll now have attached it to this new diversity gene segment here, which is D2 now. And we'll have attached D2 to our new chosen joining gene segment here. We'll then have all the other joining gene segments that follow it, and then our second constant gene segment here. So this is the second constant gene segment. So let me now just colour these in. So here is our second diversity gene segment, there in yellow. Here is our chosen joining gene segment from the second cluster of joining gene segments, and then it will continue on all the way up to, well, we'll have all the other remaining joining gene segments after that. And then finally, after all of those joining gene segments are through, we've got our second constant gene segment here. Right, so that's what would happen if the first VDJ recombination went wrong. We can now have a second attempt at it, and now if this went right, you can imagine what would happen with regards to transcription and then translation. You transcribe from here, you'd get a piece of mRNA that actually looks exactly like this. So the picture for the mRNA would look exactly like this. We have our chosen variable. This time this diversity portion corresponds to the second diversity gene segment. Here's our chosen joining gene segment, and then we'll have all the other joining gene segments after that, and now this is the bit that corresponds to the second constant gene segment, and then we'll splice all of that out to get a mature mRNA, which would then code for our beta chain. Okay, so that gives you a second shot at it, basically, if the first shot goes incorrectly. So this is the order in which you'd actually go through this. You would start off with one of your beta chain loci on one of your chromosome 7s, and you try to make a functional beta chain from that. And you get 2 shots with that single beta chain locus. If both of those fail, then you move on to the other beta chain locus on the other chromosome, and then you would again have 2 shots with that beta chain locus. And if all 4 of those shots fail, then of course the lymphoid progenitor cell would have to commit apoptosis. Okay, right, so hopefully what I have now managed to communicate to you is the fact that these developing lymphoid progenitor cells they have to design an alpha chain, they have to design a beta chain, and the variable portion of their alpha chain and the variable portion of their beta chain will be unique to that lymphoid progenitor cell. So all the different lymphoid progenitor cells are going to come up with their own variable domains and for the alpha chain and the beta chain, and therefore they're overall going to come up with their own T-cell receptor. Now they will produce loads of copies of this design of T-cell receptor. So once they've recombined the DNA, they now use these genes, these recombined alpha chain loci and beta chain loci, alpha chain locus and beta chain locus, to actually produce loads of copies of the alpha chain, loads of copies of beta chain, which are all identical, and they put loads of T-cell receptors of this specific design that they've created on their surface. Okay, so let me now continue the story on. So let me draw a picture of the thymus gland here again. So here is the capsule with the interlobular septums stretching in here. Okay, and I'll just again colour this in green here. Right, so remember our lymphoid progenitor cell was up in the subcapsular region of the thymus gland, and it's now completed this process of creating its T-cell receptor, and I'm now going to draw it with a T-cell receptor on its surface. So here, this is going to be our lymphoid progenitor cell here, and now on its surface, this is going to represent the T-cell receptor. So I've changed the picture that I've drawn for the T-cell receptor. Previously I had a much fancier picture of the T-cell receptor. Now I'm going to show it in this way, and this portion up here where I've got this groove shown here, this is going to be the variable portion. So this top portion is the variable portion. So half of it's made up of the alpha chain, half's made up of the beta chain. That would be the division. So here's the alpha chain, here's the beta chain. This top bit is the variable domain of each of them, and together they make the variable domain of the T-cell receptor, which is the portion that's actually going to be unique to an antigen fragment. Okay, so this is my new picture then, a simple picture for a T-cell receptor, and I know it looks very like an MHC molecule, but never mind. So here, this is going to be my T-cell receptor on my developing lymphoid progenitor cell, and now what's going to happen is once it's designed its T-cell receptor and it started expressing its unique T-cell receptor on its surface, it's going to also express two really important molecules on its surface as well, which I'm going to draw here. So it's going to express a molecule known as CD4, and it's going to express another molecule known as CD8. So it's going to put both of these molecules on its surface. Now those of you who know something about the adaptive immune system will recognize these names. These are really important molecules. CD4 and CD8 are very famous molecules as far as immunologists are concerned. And you will know that T-cells, mature T-cells, usually only have one of these molecules on their surface. So either they'll have CD4 on their surface and there'll be a helper T-cell, or they'll have CD8 on their surface and there'll be a cytotoxic T-cell. These developing T-cells in the thymus, temporarily, they get both CD4 and CD8 on their surface. So this cell that I've now got here, this is no longer going to be called a lymphoid progenitor cell. Now that it's designed its own T-cell receptor and it's got CD4 and CD8 expressed on its surface, it's going to be called a double positive, or a DP thymocyte. So a cell of the thymus gland here. So the DP stands for double positive. Now why is it called that? Because it's positive for both CD4 and CD8. It has CD4 on its surface and CD8 on its surface, and most T cells do not have both CD4 and CD8 on their surface. It's one or the other in mature T cells. So that's why we're stressing this fact, because it's very interesting that they've got both of them on their surface. Very few cells have both CD4 and CD8 on their surface. Okay, so to summarize where we've got to so far, the lymphoid progenitor cell has come up to the subcapsular region. It has designed its own unique T-cell receptor that has the tip portion unique to that cell. Okay, it has its own T-cell receptor, and hopefully this T-cell receptor is going to be able to bind to MHC complexes with peptide fragments in, and because they're all unique, well, because it's got its own unique T-cell receptor, it will bind and recognize a specific peptide fragment with a specific MHC molecule. Okay, it's also now got both CD4 and CD8 on its surface, and this is going to be part of its development. We'll see the importance of these molecules later on. If you don't recognize these molecules, don't worry, you will understand how important they are later on. So what's now going to happen? Well, we've just created this new cell that is going hopefully to become a mature T-cell. It's got this T-cell receptor that it's just created in this way that we've now spent a lot of time studying. What we need to make sure before we actually let this cell go loose into the bloodstream and become a T-cell is that this T-cell receptor firstly is actually useful for something. We need to make sure that it's actually capable of binding to peptide MHC complexes, because that's what has to happen. T-cells are activated by their T-cell receptors binding to MHC peptide fragment complexes presented to them by antigen-presenting cells. If this T-cell receptor can't bind to peptide MHC complexes, then it's useless, i.e. this double positive thymocyte that we've produced is useless and it's going to have to go. Secondly, we want to make sure that it's not going to bind to too strongly MHC complexes which have peptide fragments that are from proteins of your own body, because that could then possibly lead to an autoimmune adaptive immune response, and that would be very dangerous. So we need to make sure that this T-cell receptor firstly has some function at all, and secondly that its function isn't going to lead to autoimmune responses. So what now needs to occur is a selection process. We need to check that this structure that we have produced, this cell that we've produced with its own T-cell receptor, isn't, well, firstly is going to be actually useful for something, and secondly isn't going to be dangerous to the body. So what's now going to occur is a selection process, and we will study this selection process in detail starting in the next video.